Hello. In this recording, I'm going to talk about transcription, initiation, and gene expression. So we said that a gene is expressed when it's transcribed into mRNA, and when a gene is transcribed into mRNA, mRNA gets translated into protein. So gene is again, a gene is expressed when mRNA is produced or protein is produced. So let's look at the process of transcription, and specifically we are looking at transcription initiation. So I'm going to draw our DNA. So this is like DNA, and we will look at the anatomy of a gene. Okay, so this is a grid. Imagine that this is our genome, our DNA, and it's measured in base pairs. So this is base pairs. And so suppose somewhere here we will label the direction of this strand 5 prime to 3 prime. And you know that DNA is double-stranded, but for the simplicity's sake, I'm not going to draw the lower strand. I'm just drawing the top strand. Okay, so somewhere here, we have something called the TSS. So this is, first of all, this is going to be our gene right here. So this is gene. And the gene is made of both coding sequences known as exons and non-coding sequences known as introns. Okay. And okay, so this is our gene. And somewhere here we have a site called TSS. Use plain text to label this transcription initiation site. And +1. So this is where the first nucleotide occurs that will be transcribed in mRNA. So suppose here this is a C right here. So when RNA polymerase enzyme comes in, so RNA polymerase can also be called RNA pol for short. Okay, so this is RNA polymerase, this is our enzyme, and it's going to transcribe our mRNA. So the first nucleotide which it's going to put in is going to be a C. So this is the +1. And remember, mRNA always goes 5' to 3' direction, so it's identical to the top strand. It uses the bottom strand as the template. So in the bottom strand of the DNA, it says a G. So if we were to draw, the bottom strand, which is here, there would be a G right here, okay, which matches up with the C, but we won't get into that. Okay, so the next nucleotide in the strand is gonna be at +2 base. Our strand got erased, okay. So the next nucleotide is going to be +2, and suppose it's a T, so then there's the enzyme will fill in the T, and the next one is +3, and suppose that's an A, so it fills in an A. So, and then it starts, basically it makes this mRNA strand. Okay. So again, this is our gene. We have the transcription start site, and it starts the enzyme RNA polymerase makes the mRNA. Somewhere in the gene, there's an ATG. So ATG is known as translation start site. is known as translation start site. And what this means is that when this mRNA is going to be translated, it's going to, ATG is going to be used as the start codon. So in the mRNA, it's going to be a UG because T's get replaced with U's. Right? Okay, let's just erase everything so we can just redraw it again. So um, here's our DNA strand, DNA strand in base pairs, uh, and this is our gene sequence here, and we have the TSS, so let's change the color of student. So here's our +1 nucleotide transcription start site, and somewhere here is ATG, which is our translation start site. And so next to the gene at the 5' end, so remember this is our 5' end and this is our 3' end. At the 5' end next to the gene is a sequence called a promoter sequence. So right here is a sequence called promoter. Promoter sequence is located, you know, at next to the gene at the 5' end of the gene. And we obviously base pairs cannot be negative because they're all base pairs. But in molecular biology, we just decide that everything starting from +1 is positive base pairs, and we'll say that the promoter is like negative. So it's located at -away from the transcription start site. Okay. So promoter is a non-coding DNA sequence. It's a regulatory sequence, and it serves as a landing site for RNA polymerase and general transcription factor. Okay, so we said that the promoter is a landing site for RNA polymerase. It's a regulatory site. Let's actually make a table of all the important elements in transcription. And in the table, we will have two columns. One is going to be DNA elements, and DNA elements are known as cis factors. So cis means that they are located in the same place, meaning that they are not moving around. Okay. And on the other end, we will have something called trans factors. On the other end, we want to write down DNA elements that I just mentioned. So one of them is gene, and we said that the gene has the following elements in the TSS, which is the transcription start site. It has the AT, which is the translation start site. And it has things like introns, exons. Another thing that I mentioned, there is also a promoter sequence. So promoter is a landing site for general transcription factors and RNA polymerase. Now there are several, promoter is also regulatory elements. Regulatory. Um, so there's two types of promoter. There's a proximal promoter, which is located about 1,000 bases away from the gene, and then there's the distal element called the or a distal promoter, which is located far away from the gene. So we must distinguish between proximal promoter located about 1,000, or we'll just say minus 1,000 base pairs away from the gene. 1,000 bases. Uh, well, it's not really away, but it's this about what its whole length is about 1,000 base pairs, and it's next to the gene. We'll say next to the gene. And then there's another element called enhancer element, which is also known as a distal presence of bases, base pairs away from the. So what happens is that protein transcription, which I talked about in the previous lecture, I suggest you watch that video where I explain what transcription factors. So transcription factors are proteins that have these transcription factors have specific function. They can bind to specific DNA sequences, and they can also interact with proteins. Transcription factors have something called the DNA binding domain that allows them to bind to DNA sequences. For example, a transcription factor RANX2 prefers to bind to DNA sequence TNNTG. Be any nucleotide. So let's write this down here. So we have something called general transcription factor, and their names are TF to I, or TF to A, B, TF to B, TF to C, TF to I, because I studied that specific protein. So TF to I acts as a general transcription factor. And what they do is they bind to the proximal promoter, and they attack and they stabilize it. And then you have other transcription factors known as activators. So let's just write this down here. So we have transcription factors, and they come in two flavors: activators and repressors. Bind to enhancer element and help, and they interact with general transcription factors and help to further stabilize RNAs. They help to activators increase the rate of transcription. What is a repressor? Repressors interfere with activators. So suppose if an activator wants to bind to a certain portion of the promoter, and the repressor protein is sitting there, it's preventing the activator from binding to that site. And continuing, let's draw this really fast. So here we have our DNA. 5 to prime, 5 prime to 3 prime. And here's our gene. Next to it is our promoter sequence, which we draw with this arrow because that's just a custom in molecular biology, and what it tells you that basically the gene is being transcribed. Okay, so this is our distal promoter, 1,000 base pairs. In the promoter, there's something called Tata sequence, which is just made of TA repeats. So it's Tata, like Tata, I'll see you soon, Tata. Okay, and so general transcription factors bind to this lovely Tata box. So we have our protein called, they bind to the Tata box, and we will give them names. So you get the idea. So this is our general transcription factors. They bind to the Tata box because they recognize this specific sequence, TA, TA, TA. This is the kind of sequence they like. They don't like, but the DNA binding domain recognizes that sequence, and then they recruit RNA polymerase and enzyme. So here we have, let me make it, this is RNA. Okay. And RNA polymerase is gonna make a default amount of mRNA. Gonna make a default amount of mRNA. So here it's making in gray, this is our mRNA. And it's making that, okay? Now, far away from the proximal promoter is a distal element called enhancer element. It's right here. An enhancer element is another regulatory sequence that attracts other type of transcription factors, such as, for example, an activator. I'm going to make an example. There's a transcription factor that acts as an activator of osteocalcin gene transcription, and it's called RUNX2. It's very important in bone development, and it helps activate the expression of a gene called osteocalcin. So RUNX2 binds to the enhancer. So here we have RUNX2 comes in here. It binds and it kind of loops out, and DNA loops out, and RUNX2 interacts. It interacts with the general transcription factors and helps to stabilize this RNA polymerases and increases the rate of transcription. RUNX2 binds to a specific sequence called an E-box. It's a sequence called an E-box. And E-box sequence has this particular, it's a particular motif TANTG, where N can be any nucleotide. So RUNX2 likes when it sees TANTG, binds to, there's an activator that's supposed to bind promoter, but instead of the activator, there's a repressor protein. So this repressor is sitting on the promoter, and it's preventing the activator protein from coming in. So there's the activator. Activator wants to bind here, but repressor is not letting it bind to the sequence. As a result, it cannot activate gene expression from this gene. It cannot help to recruit RNA polymerase. So as a result, this gene is turned off. So in biology, molecular biology, we say when the gene is turned on, you usually have like an arrow here, but when it's turned off, it's like a line like this, showing that it's turned off. Okay, so let me erase this here. So yes, if it's turned off, it looks like just a line like.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex, able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. The activator proteins are not shown.
Tips for understanding transcription gene expression mechanism. First, review the handout with the definitions included in the folder for Monday's asynchronous assignment. The handout is provided in the folder. It would be useful to have the handout open while watching this video. Number two, if you have a case where a patient has lower levels of gene expression or higher levels of gene expression, this might be due to an issue occurring during transcription. So on the test, if you have a case where a patient has lower gene expression or higher gene expression, this might be due to some issue affecting the promoter, the enhancer, or the transcription factors. Number three, for example, if there is a mutation in a promoter, it won't allow transcription factors bind there. It would not allow them to recognize their specific binding site. Therefore, this might result in a lower rate of transcription. Therefore, less mRNA would be produced. Gene expression would be down-regulated. Number four, on the other hand, suppose there is a mutation that creates an alternative splicing site for an activator, a stronger enhancer. Hence, there would be more transcription of that gene and mRNA levels would be higher. Hence, gene expression would be upregulated. This sometimes happens in cancer where a certain oncogene is highly expressed. For example, BCL2 gene is an oncogene which is often overexpressed and upregulated in cancer. BCL2 protein prevents the cells from committing apoptosis or cell death, thus allowing cancer cells to survive. So it's very beneficial for a cancer cell to overexpress BCL2 gene. Also, estrogen receptor is often overexpressed in breast cancer. There could be multiple copies of enhancer elements or promoters which can make gene expression for estrogen receptor stronger and upregulate that specific gene, and this would drive breast cancer, make it more aggressive. Okay, thank you for listening to this lecture, and there will be an assignment that goes with it, with all of these videos that you watched. Thank you for watching the videos. Okay, bye.